And then we have peripheral proteins. Peripheral proteins are going to be proteins that are on the periphery. Remember that that means on the edges of something or on the sides. And so you can see peripheral proteins are only going to be on one side. Peripheral proteins only on one side. And glycocalyx, this is where we left off last time. Glycocalyx refers to the superficial or the sugar recognition molecules. These are carbohydrates, as we learned about, the sugars that we learned, and they form a profile around every single cell. And your white blood cells are always investigating these tissues, or as part of these surface molecules, to see if the cells are healthy, to see if the cells are infected, to see if the cell is cancerous, and if anything's wrong with that sugar profile, which is essentially like if you're thinking about medieval times and every single castle has its flags, its banners to represent the house of whatever, the castle, that is going to be what you have to see, these glycocalyxes. And so this word itself can seem a little confusing because it's not just one of these. Just this isn't a glycocalyx. It can be, but it really refers to the entire profile, the entire sugar profile on the surface of cells. And again, this is how they identify, the cells in the body identify each other. And so this phospholipid bilayer has our hydrophobic tails, water-fearing, hydrophilic heads, which love water, and they have embedded cholesterol, which helps stiffen the plasma membrane. And so depending on how much cholesterol you have on that membrane will determine how stiff that plasma membrane is. And that's really going to be a very specific situation as to why you would see it on some cells and not other cells. For us, we just need to know that cholesterol stiffens the plasma membrane. All right. And so here's that note to imply, like I mentioned before, that sometimes integral proteins are called transmembrane proteins. That's why it's more perfect if you were just to like cross this off, or you can just consider it a property of integral proteins. And so that's what this is here for. Integral proteins are integrated into the cell membrane and also connect to the cytoskeleton. And sometimes if they go through the entire membrane, meaning that they go from this side to this side, they're considered transmembrane. So it's sort of an adjective to describe a protein. And they're capable of transporting substances. As we look at this one here, for example, this one you can see is just this open channel here, which allows materials to enter into the cell as necessary. And then peripheral proteins are going to be proteins that are not going to be found embedded within the plasma membrane or on both halves, but really they're only going to be found on one side. And so that's why we say attached to the cell membrane, but it's either going to be on the inside or the outside. And don't worry about this extra stuff. I just really want you to be able to know the characteristics and use the word to help you. Peripheral, right? The outside. This is extra stuff that you don't need right now. And so with that, I also mentioned last class that we're going to limit it. Just know that glycocalyx is the sugar profile on the surface of cells. And all of this here, you're free to get rid of it. But just keep in mind that these sugar profiles, these sugar molecules, can be anchored to other proteins. They can be anchored to lipids. And so this segment was just really about understanding the naming system around that. So if it's a carbohydrate attached to a protein, then it's a proteoglycan. And so if it's a lipid attached to a lipid, then it's a glycolipid. With this one, we've already explored, and we kind of have to know what a glycolipid is. But we're going to keep it light for this segment, and you don't have to worry about really being able to recognize this. You should already know what a glycolipid is in general. And so, do not forget, a glycocalyx helps with cell recognition, helps bind surfaces to other cells or other hormones. It binds things on the surface, and it also serves as a lubrication. In other words, you're not really getting direct contact all the time with other cells. You're not really rubbing against the plasma membrane. Normally, they just, like when you think about a airplane or a boat really going onto its shore or onto its, what do you call it? What do you call a place where you park a boat? A dock, it's like a segment where other cells will dock with these molecules, and they're not really rubbing and crashing into each other. And so sometimes you'll see it characterized, or as a function, will be characterized as lubricating the cell surface. Lubricating the cell surface. And so, these are the functions of our plasma membrane. They allow for physical isolation. They regulate what goes in and out by actually absorbing, or placing things inside the cell. We'll learn that in a bit right now. They recognize of each other through that sensitivity. They recognize the environment, the fluid outside. They're capable of actually adjusting to whatever is on the outside of their plasma membrane. We can see that some of them proteins, some of those proteins are anchored to the cytoskeleton, and so they are going to be, they're going to be anchored to the cytoplasm, to the cytoskeleton, and thus helping with the structure of the cell. And the lipid bilayer separates an inside from an outside of the cell. And proteins carry out the function. Don't forget that proteins are our molecular machines that carry out functions. Now this table, the ones that I really want you to wrap your heads around today are carrier proteins and channel proteins. And then lastly, the receptor proteins. These other three are a little bit complex, and we're going to see specific examples as we get to it, so you won't have to worry about it for the exam. And so these are the definitions of the three that we're about to see, and then we're going to see some instances of them. And I'd rather just start there instead of with that part. I'll come back to this very thing after this very segment. And so, now, for this next segment, the cytoskeleton, we have to evaluate some terms around the cytoskeleton, some things you already know. For example, we know that they're going to be made out of protein, so that's what this line indicates. It provides an internal protein framework, functions as the cell skeleton. This will increase the overall strength of the entire cell and allow it to be flexible. It can increase in size and decrease in size without really necessarily damaging it. And the components of the cytoskeleton go as follows: microfilaments, intermediate filaments, and microtubules. And if you were to actually place them on a scale and compare them to each other, it's small, medium, large. So smaller, intermediate, and the largest. And we'll see why that matters more into the semester and a little bit right now. So make sure you get that now. And so, starting with our microvilli, these are the famous microvilli we keep talking about in lab. They're going to be characterized always as finger-shaped extensions. Their main purpose is to absorb on the surface of cells. Absorb on the surface of cells. And they are made out of microfilaments. Let the micro remind you of that. And with that in mind, let's look at microfilaments. You just go here to the right a little bit. And so microfilaments are exactly what a skeleton does for the human body, is that they provide structure, in particular to the microvilli, and that's the one we're going to hold on to for our class, and typically composed of actin. And this is something that we're going to return back to in muscles, so you're free to cross this off for our exam. And if you look at this little image of our microvilli, you can see our microfilaments are inside, providing that rigidity. But still, on this surface, you would still see, if we zoomed in, you would still be seeing a phospholipid bilayer with all those proteins and a glycocalyx on the surface of those cells. This particular cell is responsible for absorbing substances, and these finger-like projections help with increasing surface area, which is an important biological thing to understand. For example, if we just look at this segment here, this open one, and if we pretend that it didn't exist, it would be closed off over here. You would be missing out on a little bit of surface space, because if you had cells here, and what cells typically do you find on the epithelial surface of the digestive tract? Simple columnar. So what you would be seeing here are columnar cells. Columnar cells. Like, it's a little hard to draw. Let me see if I can draw it in just for the sake of example. And you don't have to do this. It's just so you can understand. And we'll get into this a little more in detail in a bit. But this is where you would find, this looks more cuboidal, but still, columnar cells. And you can see that all of it would be completely made out of columnar cells. But if we pretend that it didn't exist for a second, you would see that we would only be able to place a few columnar cells just in that little surface. But by increasing or creating this elongated shape, we increase the surface area. And so the whole point of this little talk is to get you to remember microvilli absorb, cilia move mucus, and also that we find columnar cells. This is an example of where we would find columnar cells, typically on villi. And I wish I hadn't said that because I just made a mistake. Because this is an actual cell, and there's a complication that even I just fooled myself with, and I really don't want to confuse you all anymore. I take back everything I said. All I need to say now is this is our columnar cell. This would be an example of a columnar cell with a plasma membrane. So this would have the same membrane that we saw in our previous photo. This is found on this. So I have to walk back that few seconds. Hopefully I can edit that out a little bit later. But otherwise, this is the plasma membrane. So you still have a plasma membrane here on the surface, but this helps increase surface area. And I'll talk to you about what I just mentioned is applicable, but a little bit further down the line. And so with that, then we have terminal web, like the terminal end of an airport usually means the end. And so you'll notice that our picture here, our diagram has this web, and it's where all these microfilaments terminate, meaning that they're all, and even more simpler, that's where they anchor to. They're anchored to this terminal web. And the intermediate filaments now is just simply going to be the strongest and most durable cytoskeleton element. And again, just because it's the intermediate size doesn't mean it's not very strong. But again, intermediate filaments are the strongest, most durable part of the cytoskeleton. And so, our next one is microtubules. These microtubules, and I really want you to understand microtubules are going to be found in our cilia. So you can see microfilaments and microvilli and cilia are going to be these long, slender structures that help move mucus, slender extensions, and they are considered motile. This word means move, they move, mobility. That they possess mobility, which should make sense to all of us by now, that cilia are stroking, and so that stroke is referred to as a power stroke. And that's simply just, again, a little cilia just moving, whipping, and then moving anything on its surface along wherever direction it strokes towards. And then just going back into that position is just called a return stroke. And so you can see that our microtubules make up our cilia, and that's all we really need to add to that. We're just going to add microtubules, make them up. And this we can cross off when you say microtubules. Just remember it's made out of microtubules, and that's good enough for me. And then the last thing, you're free to cross this off. I'm trying to cut our material since it is summer and it is a little crazy. All right. And that's it for this packet. Let's go on to our next packet. And so in Latin, exo means outer or outside, and endo means inside. And then cytosis is the process of going in or out of a cell. So exo means to go out the cell, inside means to go in, and the word diffusion is a very complex word to mean something very simple. If right now somebody I used this example in lab the other day, but if someone placed a hamburger right here on my desk, even though the molecules of that hamburger are right here, eventually those little hamburger molecules, which are the proteins, the fats, the carbohydrates, in that heat, they're dissolving in the air, they're flying into the atmosphere, and then they end up back there. And you can smell it, even though the burger never went over there. That's what diffusion is. It's a complex word to mean one thing going from here to there. And the more precise definition is going from high to low. You'll hear this again in the future in some form of physio. High to low. And doesn't that make sense if you place, this is supposed to be a hamburger, but if the hamburger, the molecules, is right here, it's very high. Wherever you place the hamburger, it's very high. And then those molecules fly all the way to the end of the room where it's low, where you're all sitting in the very back. And so that's all diffusion is really about. It's just movement from high concentration to a low concentration. And that is all diffusion means to us. Now, where things get iffy with diffusion is that if you, and this is going to be a very rough example, but it'll be enough to understand, that if I were to put a beaker, and I was to put a membrane, meaning something that has pores in it. And let's say that I put some molecule, like, doesn't matter, just a molecule. Could be salt or something. We can assume that after a certain amount of time, that these molecules are going to go and make their way to the other side. They're going to diffuse to a place from high to low. Right? There's nothing different about it. The only difference is that there's a membrane now, and this causes a very unique pattern of movement that we're going to see now. If we give it enough time, what you can imagine is that even though you have this membrane, is that these molecules will be evenly distributed on both sides. 1, 2, 3, 4, 5, 6, 7, 8. 8. So 1, 2, 3, 4, 4, just for simplicity's sake. So we started off with 8 on this side. Right, and then after you give it enough time and they can go through that membrane, you'll see that we have eventually 4 and 4, and they're going to be something in what we call equilibrium, which means that it's not always going to be 4 and 4, because molecules are always moving. That means that when one goes this way, one will come back. But you will always have, generally speaking, a net of 4 on one side, 4 on the other side, and we refer to that as being in equilibrium. That is what diffusion looks like when you have a membrane, right? It's easy with our hamburger example, but this membrane adds that little bit of complexity to it. And it'll really matter when we talk about blood and water regulation. Yes?
Is the act of it going through the membrane, is that osmosis?
That would be if water is moving through. When diffusion is the same thing, osmosis is the same thing as diffusion. The only difference is that when we're talking about the movement of water, then it's osmosis, but it is no different. Water can technically be more abundant on one side than on the other side, but we get into that complication a little bit further down when we talk about water movement around red blood cells. But otherwise, today we just keep it simple. We really just want to know what diffusion is and that it's defined as going from high to low, because it'll help us understand some things that are going on the surface. So I'm going to quickly jump to, never mind, let's stay here. All right, so endocytosis and exocytosis is about vesicular transport within a cell, which means the formation of vesicles, or vesicles are somehow interacting with other vesicles, and we already have some premise behind it. So without reading all this text, I'm going to just say things simply and then get into the complication. On the surface, this red line here is our plasma membrane, our bilayer. It has those receptors on the surface, and when a certain molecule, which for us, ligand means the same thing as just a molecule that binds to a receptor. That's it. Because you have to make the distinction, because technically these protein machines are made of amino acids. They are also molecules. So it's not, if you say one molecule finds that molecule, the language would just be so confusing. But otherwise, this molecular machine, this protein, is going to be our protein receptor, a protein receptor. The word ligand in this case is going to be something that binds to a receptor. So I need to clarify this. Molecule that binds, let's just rewrite it. Molecule that binds to receptor. Too thick. Molecule that binds to receptor. And just as simply, the receptors that bind to these molecules are called ligand receptors. Nice and easy. So the only adaptation we need to add is ligands, the word ligand. It means a molecule that binds to a receptor. And so, if enough ligands bind to the receptors, what we see is that they will eventually, or these proteins in union with other little proteins, will then finally pinch a vesicle inside of the cell. When enough receptors bind, they cause the structural changes, and then it recruits proteins at times. That's what the role of the ligands are. And then they'll form a vesicle that goes inside the cell, and that vesicle will now trap all these molecules on the inside, all those ligands that the body that the cell wanted, right? So these ligands are in the extracellular fluid, in the extracellular matrix, and they want to go into the cytoplasm. And so when they enter still in this vesicle. This vesicle is made out of lipids. As we've seen, lipids have this property that when they're together, they can form a center that keeps everything away from the outside. And so they store this ligand temporarily. And then our lysosomes, remember that our lysosomes are filled with digestive enzymes to help break things apart. And so they combine with it, because remember, this lysosome is also a some, implying that it's made out of lipids. And so if this vesicle is made out of lipids, because it's the same color as the plasma membrane, if you'll notice here, this is the same thing as the plasma membrane. It's a phospholipid bilayer. It's a lipid made out of lipids. But so is this. And so they can come together and combine because they're made of almost the same exact thing. And so they just sort of fuse together, bless you. And then these digestive enzymes can go in and help. And they'll do a variety of functions, some complexity we don't need. But you can see here that they will break the ligands off, and now these ligands can be utilized inside of the cell in one way or another. And then finally, when our lysosome has nothing to actually work upon, then it detaches. And this, by itself, without the ligands, can't stay or sustain its vesicle. And so it returns back and becomes part of the plasma membrane. So we've only seen in one direction, right? Things finally go onto the surface, form a vesicle, digest. The second everything's digested, that vesicle comes right back. This process is referred to as endocytosis. Endocytosis. This explanation here, saturated, means to be overwhelmed or to be full of, to be fully covered. But overwhelmed, I think, is the best or simplest way to understand it. That if enough ligands bind to these receptors, it overwhelms it. It saturates it. That's the fancy way of saying it. And this will cause the pinching of that vesicle. But this vesicle is not simply just called a vesicle, because since it's going in, endo, and some, remember, like lysosome, peroxisome, usually implies a vesicle. So this vesicle, or endosome, is a vesicle going inside. Going in. These little clathrin-coated pits, they are responsible for, when they detect saturation, they're what sort of cause this enveloping or this pinching that will eventually seal it. And once it's made and inside the cytoplasm, they release and they go look for more segments that are saturated. And we refer to these vesicles, these endosomes, when they have these clathrin-coated vesicles, as clathrin-coated vesicles. And after that, the same endosome fuses with a lysosome, and it can digest substances. The ligands will be released, and we don't know what this could be. This could be a variety of things, a variety of components. It could be nutrients, it could be hormones, it could be absolutely anything. But their goal was to go from the surface to go inside. But you'll notice that they didn't just diffuse through. They didn't just go from high to low. Just because this membrane, even though it is somewhat permeable, it doesn't just allow random input of things. The cell can take things in faster if it wants to, and so that's kind of a little bit of a take home. We'll elaborate on that a little bit further down the line. And so now we're inside, and then the vesicle did its job. It doesn't have clathrin coated pits to sustain or to sort of trigger this lysosome fusion, and it just returns back to the surface, and that is what we refer to as going. When we're going back in this direction, we're just going to become back to the plasma membrane. Ignore this. This is something I really wish wasn't here. But otherwise, it just means this vesicle is just heading to the surface. That's it. You can actually cross this out for now. Yeah, cross it out. Because when we see it at the very bottom, you can see that exocytosis is simply when a vesicle is actually going to release substances to the surface. And that's why I really don't like it there, because I really want you to know exocytosis as the movement of material to the outside world. So I don't like this being characterized as exocytosis. I actually don't believe it is exocytosis. I do think that's just it's an incomplete image, or the image is part of another bigger image that's got cut off. But otherwise, this segment here is exactly what it looks like. Here's our molecules being made by the cell, products made by the cell. This could be mucus, proteins. And so these proteins will be stored, say, inside of the vesicles, and then the vesicles or lipids, whatever is going to be secreted, the mucus is going to make its way to the plasma membrane, and the contents will be released to the outside. And that's what we're going to know exocytosis is. And you can get rid of all this. Get rid of all this. If you copied it down, sorry. But otherwise, exocytosis is just the removal or sending things to the surface and releasing it out of into the extracellular matrix. Ask away.
Yeah. So like exocytosis and endocytosis, they are not occurring in the same cycle. Is that what you understand?
This textbook characterizes the movement of direction of the vesicle as endo or exo. So if it's going into the cell, endo. If it's going out, exo. But otherwise,
Yeah, it's because as that picture, so it seems like they are in the same circle. But you say it's not exactly right.
I don't like no, I think it's more of a particular taste, because you're never going to see, I don't think you're ever going to see vesicles that were originally entered through endocytosis be classified as exocytosis, because exocytosis is the movement of materials outside, endocytosis is the movement of materials inside. But you'll notice that this vesicle, there's nothing inside. And so this is just a complete circle, and I think this textbook just classifies the movement of vesicles in and movements of vesicles out. So even this, I think this textbook classifies it as exocytosis. And so I will say you can hold on to both concepts, both perceptions, but I've never really heard exocytosis being the complete circle of the plasma membrane just returning back, but it could just be something that I'm forgetting. But exocytosis to me, functionally, is just that when contents are released to the extracellular environment. Yes, but that's a good question. Otherwise, but and even if I'll keep it consistent with the publisher, I don't ask for anything that's going to say, oh, the vesicle back to exocytosis, but if you ever see it again, then we can say we saw it, and that's something for me to actually elaborate on my own. All right. And so try to remember diffusion is going from high to low. So a word we have to adopt, which seems a little intimidating, is the word concentration gradient. As it's in bold right there for you as well. Concentration gradient. And all it means, high to low. That's a concentration gradient, because you have a certain concentration here, and then you have a concentration here. So a gradient is sort of like degrees of something. Like this is a degree of lines, right? There's a lot here, and then the gradient gets thinner and thinner and thinner, right? That's what the gradient is. It's sort of a difference between point A and point B. And so when we say high to low, that is a concentration gradient. Even if we look at this simple beaker example, the water right now, low, and this little sugar cube, it could be anything, a cube of little molecules that dissolves easily, and low, and then this is where it's going to be very high. And if you were to place it, and I'll rewrite this right now. And so you can see that if you place it inside of this beaker, this is the area of high, and I'm going to return it now, high. You can see high, And then if you give it enough time, it goes from high to low, meaning this area still has water, doesn't have any of these molecules. They're diffusing to areas that are low. So don't be intimidated by the word diffusion. It just means going from high to low. And the word concentration gradient. If you look at this, this is what the H symbolizes for high. This is what this L symbolizes. And in your little photo, you can see this little bit of dissipation or a little bit of dissolving of those, whatever was placed in there, the molecules. They're sort of traveling through or diffusing through, but they're still high and a medium and then low. So, but this is, again, what a concentration gradient is. You're just really going from high to low. And then eventually, with enough time, you will see that it spreads throughout the entire beaker, that the molecules will be equally distributed throughout. And the same thing is occurring even when we place, if we were to place a membrane here and I were to put the molecule, that little sugar cube there, eventually you can expect that it'll travel to the other side as long as this membrane allows it, that it'll travel to the other side, and then finally it'll look just like this beaker right now. It'll be evenly distributed throughout on both sides. And so, the scientific language around it is whenever you have something that's going to be placed, something that's going to be placed into it or into a solution, into water typically. It's always going to be water unless you're taking like Ochem or something. But otherwise, whenever you're placing something in water, something that can dissolve a solid, right? We call that a solute. A solute. And then we consider this the solvent, because it dissolves, right? Dissolves the solute. And so when no more movement is occurring, no net movement, we refer to that as equilibrium. Equilibrium, right? Because if we're looking at our last beaker, there's no different concentration. It is the same here as it is here. Here, there's no high to low anymore. And so there's no real net movement. And that doesn't mean that the molecules aren't moving. It just means that there's no more difference. Because here, different, this one, let's use this picture. Here, different from here. Different down here, different up here. Here, still different down here, getting there, but still different up here. Here, all the same. So the molecules are just evenly moving equally, and so that's referred to as no net movement. It does not mean no movement. All molecules, especially liquids, are always in motion. Even my water bottle in here, the reason it's still a liquid is because these molecules are always just moving and moving. They're never settling like this hard material. But that's the big difference, or something that's a little hard to grasp, between no movement and no net movement. And so with that. Now, the reason this is a topic, and we're going to touch on it very lightly, and the reason it matters for the cell, because the cell, what does a cell do? A cell doesn't walk around, not all cells, right? Like only a few cells move around, but a cell's just in place. Their action, their function is how they interact with the outside world. And that's why the plasma membrane must not be seen as just a wall of separation. It has to be seen as the arm of a cell, even though it's not really an arm, but it's how it interacts with the outside world. When something's wrong with the outside world, it adjusts from the inside, but it always starts at that plasma membrane. And with that, you can have movement or diffusion through the membrane. We're going to start with this first one here. Movement across the lipid portion of the membrane. That means to be able to move through the lipid portion. Remember how I said this is not a dead wall. It sort of resembles the concept of if you're at Chuck E. Cheese and you see that ball pit, that technically if you were to reach your arm in, you can like touch all the way to the ground, and then the balls just sort of close up on your arm. Like as you place it in, all the balls sort of seal up, and then you can take it out, and then the balls fill that hole that your arm was in right now. And so that's kind of what this can do. And so if this particular material, what is it made out of? What molecule? What organic molecule? What's the entire molecule called? A lipid. So this is a lipid. And so this particular molecule, is also a lipid soluble. Lipid soluble. That means that it can dissolve in lipids. And so the entire plasma membrane is made out of lipids. And so anything that's called lipid soluble implies it can actually just go right through. Because remember that like actually likes like. And let's go back to this right here very quickly. Remember that we said that lysosomes are made out of lipids, the surface of it, right? The enclosure. So is this vesicle made out of lipids. And so like finds like, and so they kind of are capable of interacting or fusing with each other well. And so these molecules, if it's called a lipid-soluble molecule, that means that it doesn't actually need a little opening for it. It means that it can actually just go right through the lipid bilayer, the plasma membrane itself, because it's lipid-soluble, meaning it can sort of dissolve into the lipids. And so we see that some molecules can diffuse just directly through the plasma membrane. They don't need a little opening. They actually can just go from high and then go to low, that they just can move through the membrane. And they're going to try to go to reach equilibrium eventually if there's enough molecules, until eventually there's no net movement. But otherwise, they can diffuse through the membrane. So that's a form of movement that we see, or a form of diffusion. And so that's what these examples indicate, that other lipids can just go right through that plasma membrane. Molecules that are labeled as lipid soluble can go right through that membrane. And then gases. Gases such as oxygen and CO2, the famous two that we need to know. Oxygen plus CO2. And let me create a bracket because this looks like a C. There we go. And so even gases can go from high to low, as shown here, right? Soluble gases. Some movement through the cell will diffuse through channels. And that's as simple, that's as simple as like when you think about the Panama Canal, like it's a channel between one area to the other to allow boats to go through land. And that's essentially what this is, or even more easier to understand, a door, right? If you were a lipid soluble, the word lipid soluble is like saying the word wall soluble. If it's wall soluble, I'd be able to walk through like this. Like I'd just be able to go through it and then I'd be on the other side. Wall soluble, lipid soluble. A channel protein is like a door, maybe more like a tunnel, maybe a little tunnel, but otherwise it'd be an entry inside of the cell. And so that's kind of what you want to wrap your head around, is that movements can occur through the plasma membrane, or sometimes water soluble. Where's my little highlight? There we go. Water, as an example, it's a decently bigger molecule. Not that big actually. It's really small. And then so it's a small molecule that can be forced into the cell through those channels. And I say the word forced right now because that'll matter in a second. But otherwise, this is an example of our water, small molecule channel. Here's our channel protein. And this is an example of a transmembrane protein, right? Because it's on both sides. And it's integrated into the membrane. And so it can allow, we can see these little water molecules. These little water molecules, the cell will always need water for what kind of reactions? When do we usually use water? When we're talking about metabolism? Hydrolysis, right? So water is an essential component or a chemical reactant to carry out a lot of metabolic functions. And so sometimes the cell, it can't wait for it to just go through here. It just can't wait. It needs channel proteins to sort of always provide a readily source of water. Ions as well, such as the salt that we learned about, sodium or chloride, they go through these membrane channels as well. And then we have carrier molecules. These are only really for very specialized molecules. Where's my little pointer here? And so large molecules won't always fit through these small channels. And so you're always going to need either a special protein, or there's just going to be some proteins that help move it in a different way. For example, here you're seeing a little protein. Here you're seeing a little carbohydrate. And here they're sort of showing you, oh, it won't fit through the channel. So they're going to have to rely on something called a carrier protein. And let's look at one right now. And we'll come back to this little tidbit in a sec. Now, when we look at this photo, this here again is a little sugar molecule. I drew these little C's and these HOHs to show you that's the same thing as having a glucose molecule. It's the same glucose molecule. And this is an example of a carrier protein. It doesn't just go through a channel like water did, like a straight path. Instead, these proteins are machines. When there's one anchored here, then finally it detects that it's anchored, and then it'll open up. It's not, oh yes it is. It's open, it shows in this illustration. Then once it detects the glucose is bound, then it will send the glucose inside. So they function like, one, bloop, one, bloop. And that's really how they work. They don't work much like the channel proteins, which were, by comparison, just an open pathway. Right, oh, that was terrible. Yes, and so like in the previous instance, we saw they just go right through like an open channel. That's the difference between a channel protein and a carrier protein. And so, what do we need to add now? Okay. Now, there is two concepts surrounding diffusion. Diffusion is a natural phenomena of nature. Like when I put that burger here, I didn't need batteries. I didn't need to get up. I didn't need to do anything to get those molecules to go back there. I didn't have to do absolutely nothing. The universe does it. The little molecules go up, and just by chance, really, other physical forces, they're traveling, it just travels to an area of high concentration to an area of low concentration. I didn't have to do anything. I didn't have to use any energy. And so, when we see the word facilitated diffusion, this means to encourage. Dang it, I dropped my pen and made it worse. To encourage diffusion. And still, how can I explain this a little bit more easier? Let's see what pictures I have here. When we were to compare this molecule, and we're going to go ahead and use our imaginations. And if we've depended on this, right, we're going from high to low. And this molecule going from this high portion, going to this portion, can be a little ineffective, meaning that it will take forever. So if this is going to take forever, what the body or the cells can do, the cells, what they can do is that they can have a protein specific for this. So instead of relying on it to naturally diffuse through, instead it might create a very specific protein that has the shape for this molecule. And then it'll be faster for it to go through here than it would be for it to go through here. And so facilitated diffusion is just a way of increasing the speed at which things go from the extracellular side to the intracellular side. And so carrier molecules, like as we saw this one, this carrier protein is an example of that. Instead of, because some glucose molecules might actually be able, with enough force, as they're going through through the fluid in your bloodstream and in contact with the tissues, some of them might actually go through the plasma membrane, maybe, but it'd be so low that it's honestly non-consequential, not inconsequential, meaning that it wouldn't matter because it's just so slow, nothing would ever happen effectively. But these proteins are a form of facilitated diffusion because now it speeds up the rate at which things go through, which will matter when you go over GLUT and proteins, which are the ones that allow sugar to go into the cells. And that's kind of the talk about insulin and diabetes. But otherwise, insulin is sort of what encourages these carrier proteins to go to the plasma membrane, and then it forces glucose into the cells. But otherwise, without insulin, these proteins are non-existent. They're not there. Insulin makes these carrier proteins travel to the plasma membrane, and then it allows the glucose to enter the cell. That's why insulin is so important, because without it, it doesn't encourage the recruitment of these proteins that will allow sugar to enter the cell, and then the body dies, because the body needs sugar for energy. It is a fuel. And still, I still said the word diffusion. That's the key word. When I say diffusion, no energy is involved. So requires no ATP. Now, what is ATP? ATP is produced by the mitochondria, and that's the energy molecule. When we talk about energy in a cell, in the molecular processes in general, we are talking about this molecule, ATP. ATP. Right now, all we need to know it as is ATP, and this is what gives energy. Energy for chemical reactions. That's all we need to know about ATP. And it's created by the famous mitochondria. And that's why it's called the powerhouse of the cell, because it produces energy, or ATP. And diffusion, again, never needs energy. The universe encourages. The only thing that you could do is facilitate it. You could speed it up. And so if you speed it up, then technically that's all you can do, just because you create a larger opening. It can go from high to low faster. Now, active transport is the complete opposite. It is actually going to need energy, so it will use energy, requiring energy molecule, or ATP. It is independent of concentration, and that simply means it doesn't care about high to low. Remember, the concentration gradient is going from high to low when we talk about diffusion, but when we talk about active transport, in this instance, it is going from low to high, because sometimes the cells can't wait for Mother Nature to just let the molecules move through on its own accord. And so what active transport is that it uses energy and it speeds up the actual rate at which things move. And so that's why we say independent, or rather it can go from low to high. It has that capability because it uses energy. Just like the same thing with the hamburger. If I use my own bodily fuel to grab that hamburger and I go to the other side and I start waving it around, then I've delivered the molecules, and that required energy input. It required me to get up. It required me to use muscles. That means I had to use energy in order to from the food that I ate in order to carry out that process. But I made it go from high to low, or really low to high really fast, right? I can just throw it over there if I want. But otherwise, that's kind of like a very rough way to understand active transport versus passive transport. But it does the job, I think. And so an example of this are ion pumps. They will always require ATP, and it encourages the entry of sodium. It encourages the entry of calcium. It encourages the entry of potassium and magnesium. Make sure that you know what these acronyms represent. They're up here in that order for you. Sodium, potassium, calcium, and magnesium. These are all very important in the molecular processes of cells. And then there's a famous one known as the sodium-potassium pump, because it does an exchange between sodium and potassium, and it's called an ATPase because it uses ATP. And its role is to always exchange 3 sodiums for 2 potassiums. And let's use the photos here. When you look at this segment, you'll see that sodium starts off here, potassium starts up here, and then 3 will go out and 2 will go in. But it requires an input of energy. An input of energy, and that's all. This is an example that's going to come back to haunt us in nervous. And so sodium goes out, potassium goes in. And this is how the cell can do a lot of very useful functions. And with that,
Professor,
ask away.
for the exam on this, do we have to memorize the amount of sodium?
No. You do not. You just have to know what a sodium-potassium ATPase is. Yeah. And then lastly, to limit the material, because we're here at an hour, I will remove this part here, because I kind of want to just move on to tissues now. Yeah, and then I'll come back to this when we actually discuss very specific examples in in organ systems, and then we'll bring it back. But otherwise, I kind of want to move on from here. All right. Yes, because we have 1 hour left. And so the good news is, everything that you've covered, you already should know most of it by now. For example, if we start small, we learned a little bit about atoms. We learned a little about molecules, especially the organic compounds, more important, and then some inorganic compounds like some ions. Now we know a little bit more now. We know magnesium, we know calcium now, we know sodium, we know chloride. Those are all examples of ions. And then molecules are those organic compounds we talked about, lipids, carbohydrates, nucleic acids, and missing one, proteins. And all together, they combine, they're the building blocks of cells. Cells have a nucleus, they have all those little organelles, and they're capable of secreting things to the outside environment called the extracellular matrix or extracellular space. And so you can see extracellular material and fluids can be secreted, and they're regulated by the plasma membrane on the surface. And now tissues, which you all have sort of already got a preview of it. The word cells is like saying the word person. Person. And the word tissues is like saying the word people. It's like a plural form of it. And we see that there's very unique interaction dynamics between cells in the same way that there's very dynamic ways that one individual interacts with another individual. And then we saw that we are made of 4 Lego sets in a way, right? There's an error here. This one, it's already there. This one should say muscle. From the publisher, I don't know what happened there. Gotta send them a letter. Here, muscles. This should say muscle tissue. And here, epithelial tissue is going to be again on surfaces. It's going to be on internal passageways. And they also make up glands. They're capable of secreting things. Lab number 2, or later's lab, I know that we haven't gone over connective tissue yet, but for now, what we have to know is that they can store energy. They provide a framework. They produce proteins that are fibers that create a nice network for things. And sometimes they're just there to cushion things. They fill spaces up. And we'll learn specific instances a little bit into this lecture and in lab. And muscle should be all for movement in the body. We have skeletal muscle. We should be aware of that one. Cardiac muscle, we're aware of that one. And smooth muscle in our digestive tract. And then we had nervous, and all we had was the neuron and the glial cells. Those were the two in the nervous tissue. And they're responsible for conducting electrical impulses and carrying information to and from the brain. And so we're going to start with epithelial tissue. Just like in lab, they are avascular. All the nutrients that they are going to acquire come from the connective tissue that's underneath, because we're going to see that one of the things that is a connective tissue is blood. Blood itself is a connective tissue, and blood is carrying in it the plasma and the fluid. It's carrying nutrients that your body absorbed. And so these blood vessels in this connective tissue layer are going to be dropped off into these spaces, and those molecules are going to be placed into the cells, and then these cells can carry out their functions by using those nutrients. That is referred to as being avascular because they don't have blood vessels directly attached to them. That's the big difference. And so epithelia has no blood vessels. They make up your outer surfaces just like the skin. They make up internal cavities, as we learned, like the rectum, vagina, or the throat, wherever there's a lot of friction. And I don't remember what's here, so I'm going to get rid of it. And here. And then so when we have glands in the body that secrete substances, and glands are made up of epithelial cells, and we'll see some very unique ones. And I like this picture here because we can see a segue into that here. When you look at epithelia, remember the surface? What do we call this empty side? Lumen. And then this is the same picture. Here you're seeing epithelial cells. Is this, what kind of layering is this? Simple. And so you can see that as we go, this is what a gland is, even on your skin. Your skin essentially is made out of epithelial cells. It's a little more complicated because they're stacked and there's a bunch of layers. But otherwise, if you go with the microscope, you would see that there are these little pores or these ducts. So your notes might have read, oh, you find glands or cuboidal cells in the glands or in the ducts of glands. And ducts is the same word as the air ducts, just a tunnel. That's it, like a passageway. And so this is a duct. And so you can see that it's epithelial, and then it transforms into a brand new epithelia. This could be anything. This could be like, let's pretend it's stratified squamous. I know it's not stratified, but let's pretend it was stratified squamous. And then it can go stratified, and then a little duct, and on the inside, it turns into cuboidal cells. Exactly like this. Cuboidal cells and then stratified all over again. And so that's kind of the way to think about glands, is that they're continuous with epithelial, but they embed themselves, and then they're going to secrete stuff here, like sweat, as an example, and then they're going to place it onto the surface and keep your body nice and cool when it's really hot out. And so some themes like exo, right, and endo. Exocrine secretes out of the duct. That's its definition. Exocrine glands. And I'll come back to this note. This is for a little bit later. But otherwise, secrete out of the duct. That means that it just secretes substances like this. And just to touch on this, endocrine, what's the difference between endocrine and exocrine? Is that endocrine relies on blood vessels. And think about me right now as just a person on this table. And if I was doing an exocrine function, exocrine means to throw something right in front of you, just like this. The gland's right here, it secretes just right here. And it's the same thing. This is an exocrine function I just did, exocrine, just right here. But endocrine would be, is if I put this in the mailbox down there, and then it gets delivered over there, and then gets delivered, and then finally someone places it for me over here. Right? That's what exocrine is. It requires the molecule to go through the blood vessels and be delivered. Another better example, I guess actual example, your pituitary gland controls functions in the testes or the ovaries. And how does it do it from all the way up here without being connected by nerves? And so it sends hormones in the blood vessels, and then the blood vessels will eventually deliver it to the target tissue. So it didn't just secrete it and then start working. It put it in my blood vessels, and then it traveled all the way down to the target tissue. And so that's sort of the best way to think about it. It's like exocrine is just right here. Endocrine, I have to put something in the mail just for it to go where I want it to. That's kind of the big difference, the big major take home from those two, is that exocrine glands secrete out of the duct, and endocrine usually are distributed by the bloodstream. Yeah, I guess I touched on it now, but yeah. And endocrine glands are made out of epithelial glands. They're made out of epithelial tissues, which is our famous tissue that we're going to use glands as an example for a location. Anybody remember? Is there no courage in this class? Which ones create glands?
Oh,
Cuboidal cells, exactly. So cuboidal cells is an example of glands, and they're considered epithelial cells. All right, let's keep going. And awesome. A lot of this is going to sound very familiar, which is kind of nice. It's going to serve as a review. I know you have your exam tomorrow, so let's move as quickly and as efficiently as possible. Let me just grab some water. Okay. All right. So basics. Histology, study of tissues. We know this. Cells working together, tissues. We know this. This first packet that you have will cover epithelial, muscle, and neural. And then connective is on the other packet. And here, surfaces. We have two sides or polarities to cells, meaning two sides in a way. An apical surface is going to be the surface in contact with the lumen, just like we learned in lab. Nothing different to add here. Microvilli are found on the side where the lumen is. And cilia are also found on the side where the lumen is. What we're looking at here is one individual cell. We are seeing the components of a cell. We're seeing the nucleus. We're seeing the rough endoplasmic reticulum. I know it's rough because it has what? Ribosomes. And then, so what's this one then? Smooth ER. And what are these little roly-poly looking ones? And what do they produce? ATP. And so, what is this one here? Golgi apparatus or Golgi body. And it secretes vesicles, and these vesicles make their way. And if we're making our way to the surface, what process is that? Close. I don't know if I heard it right. Say one more time. Is it exo or endo? Exo. So vesicles leave the cell exocytosis. Endocytosis is when a vesicle goes into the cell. And so we're sort of seeing if a cell, the nucleus will command the cell to create some contents, maybe proteins that are destined to go to the plasma membrane. And so those proteins are made by these ribosomes. And then they're going to go to the Golgi body. And the Golgi body will deliver those proteins to the plasma membrane. And that's how we can have channel proteins, integral proteins, transmembrane proteins, carrier proteins. They arrive through the Golgi apparatus, delivering them through the vesicles. And then the smooth ER. What does the smooth ER make? Lipids. And so we know we need lipids on the plasma membrane, on the surface of the cell, all over. And so the Golgi apparatus will also deliver those lipids all the way to the actual plasma membrane. So with that, this side is our apical side. And then we have our basal side. I've only taught you basal side. Basal implies the bottom side. This word is a combination of two things that you already know, basal and lateral, meaning the bottom and the side. Bottom and the side. So you can see that the arrows, one goes to the side, one is going all the way to the bottom. So basolateral is just to sum up both these sides. But it's more practical if you learn basal as well. So basal is going to be the bottom side, bottom. And then if you just simply describe this one, it's lateral. The lateral side of the cell. So you can see that these cells do have an orientation just like a person does, right? A head, feet, like a top and a bottom in an essence. And so that creates, again, a polarity, meaning two different sides. This is going to be in contact with the external world, the external environment, and this is going to be responsible for staying anchored and also for the passage of materials to go through it as well, because they are avascular. We're not seeing any drawings of blood vessels in here. They are avascular. It doesn't exist. They get their nutrient from the connective tissue below. And then this basal side, what do we always find at the bottom of all epithelial cells? The basement membrane. And this is where you would find that same basement membrane on that basal side. Now we have to add some new words that I don't see anywhere. I see it over here. Interstitial fluid. Sorry, it's a little hard to write. I wrote it last semester. Interstitial, with an A. Interstitial. Trying to clean it up here. Interstitial fluid, or it's the same word as also as you learned it, extracellular fluid. It's the same exact word. Interstitial fluid. Because now, the reason it matters is because you'll notice that these cells, and this is like a drawing, right, to show you the difference between cilia and microvilli, but you will never see cilia and then microvilli like this. This is just an animation. It's not like this. But what is in common with this space is that if you were just thinking that they were the same cell, this fluid, they share it. They share this entire space together. Whatever's on this side will affect this one. Whatever this one secretes out of here will affect this one. So the term interstitial fluid refers to the fluid around cells. It's just a fancy word to mean fluid around tissues. Tissues. That's all interstitial fluid is. And so with that, some functions of epithelial cells are to avoid dehydration, like the skin. We saw stratified squamous does that. It protects us from friction, right? Abrasion. And again, it just serves an outer protection. Outer protection. That's the key take home from here. An outer protection. Protection from the external environment. That's better. Protection from the external environment. Oh, I guess it's already here. Provide physical protection. Just learn what's in bold to keep it simple. It controls permeability. This means that it doesn't just allow everything to go through you or out of you. And so it controls, it sort of forms a barrier. That's the best way to, that's what this word implies, a barrier. And let me get rid of this here. It provides sensation, because we're going to see that even nerve cells, they sort of allow nerve cells to embed themselves in there. But keep in mind that nerve cells are part of a different family of tissues, a different family of tissues. But again, they provide sensation. Like, for example, your skin, on the surface of your skin. Skin was the famous example we used for keratinized, stratified squamous. And you can feel things because there are nerve cells embedded in that skin. And so we refer to that as neuroepithelia because it's a combination of nervous tissue and epithelia. We know they form glands. What do glands do? Secrete stuff. And that's all we have for our functions. Don't forget, the name of the outside space is the lumen on the apical side. Microvilli are for absorption. I think you're all sick of me repeating that over and over. Cilia are going to be for the movement of mucus. You may see the word mucin on my notes or text or out in the world literature. It's the same word as mucus. As far as you're concerned right now, we'll elaborate a little bit later. And they also, this is an extra fact that I feel like we do not need. I don't want you wasting time on, so don't worry about this. And I'll remove it for next semester. All right, next page. And so this is the last real new stuff to add. The rest of it's going to seem very redundant after this little segment. And this is about how they actually stay together, right? There's not cement or glue. Right? We have to know how they actually stick together, and that's what this portion's about, and how they communicate with each other. And so these are referred to as simply epithelial attachments. And that's just characterized as attachments between cells that are next to it. That's all. Adjacent cells and adjacent tissues just means the tissues next door or the cells next door. And so. And I see our slide. You don't have to flip pages. You might have to. And so starting with hemidesmosomes, kind of going in the order that I have here. I don't know what happened with the page order, but something happened. And so hemidesmosomes simply anchor the epithelial cell to the basement membrane. Now we know what that glue is, right? That basement membrane is underneath all of these. We can get this in frame. Nice. No, a little higher up. And so here, this segment here is going to be your basement membrane. And so you can see where our hemidesmosomes exist. Remember that that's their role, is to keep it attached to the basement membrane. And something I'm going to remove, I'll remove this little fact. I think it's a little bit extra for the summer. It's made of two parts, just as an FYI. They're here for you, basal lamina and reticular lamina. If you're watching this video and it's the summer, you don't have to know it. If you're in the fall, you better memorize it right now. All right. And so with that, hemidesmosomes are going to be anchored to that basement membrane. That's it. That's all we need to know. Desmosomes sound very similar. Hemidesmosomes, desmosomes, and that's because they do the exact same thing, but they're not on the basal side, they're on the lateral sides. And so they're usually characterized as like buttons, and because they sort of make the different parts of the shirt just anchor together. And so desmosomes are firm interlocking proteins. proteins, and they are proteins on the plasma membrane. They work by having these very glue-like molecules. That's what the word adhesion means, right? Like a glue. And so cell gluing molecules. We refer to them as CAMs. And do not worry about this other bolded fact. And this is part of the reason that your skin on the surface is waterproof, because you have very tight desmosomes, or all those squamous cells that are tightly compact together. They're anchored and stuck together by desmosomes, very tightly bound together. And so, next one. Gap junctions. Believe it or not, you already learned about gap junctions. Gap junctions are going to be these proteins that create channels between two different cells. So let's look at the big picture one instead of that one. Here. Gap junctions are a form or a way of chemical communication between cells. They allow the sharing of materials between each other. They communicate with each other. Here's another view of it too, right here. You can see this is a gap junction. They're going right through here. The reason that I say you've already learned it, and you may not even be aware of it, is that you saw these discs in cardiac muscle. What were they called? Intercalated discs. Those intercalated discs, those large little bands that you were able to see, the reason you were able to see them is because they're actually gap junctions between one cell and the next cell. So they're passaging a material with each other, or electrical conduction with those gap junctions. And so what's underlined is what's most important for you all here. These proteins are an example of the transmembrane proteins I mentioned previously, and connexons. That's the name of the transmembrane proteins of gap junctions. And then adhesion belts, feel free to remove it as well. For this exam, it's fine. And then the last one we got is tight junctions. Tight junctions, all their goal is to anchor the plasma membranes together, like knitting in a way, like it knits them together, and it makes them completely waterproof in between, meaning that it doesn't just allow things to go. Like if you look at this photo here, you're seeing a cell that's been sort of, everything's been removed from the inside. You're seeing another little columnar cell here. And so these tight junctions that you're seeing sort of keep them connected, and it doesn't allow things to just start going, like liquid to go between where the gap junctions are, where the desmosomes are. It keeps them tightly bound together, at the plasma membrane end or the apical end typically, in order to make them waterproof in the lateral sides, to not allow water and solutes in between the cells. All right. And that's really the last bit of new material, I think, to add. Otherwise, the rest of this is going to sound like complete refreshing your memories for tomorrow's exam. And so we have simple squamous. We should know why it's called simple squamous by now. There are two things to add for our lecture exam where we cover this, and that is going to be mesothelia and endothelia. Mesothelia, you already know what it is. What is this cavity called around the heart? The pericardial cavity. This is made out of simple squamous. That's a location you could even use for the exam tomorrow. If I ask you for a location for simple squamous, you either use alveoli or air sacs or mesothelia, which is pretty much the cavity, a ventral cavity, right? and endothelia. This means inside the blood vessels for now. We're just going to keep it simple. The blood vessels. Remember that blood vessels also need to be able to exchange gases really easy. So the inside of blood vessels has simple squamous as well. It is only one layer, and it is a great tissue for diffusion, absorption of gases, and reduction of friction as well, because we see it here on our pericardial cavity. Remember that this cavity is about lubricating the heart as it beats. And you also find mesothelia around in these cavities, but we don't really go over this one yet, not till digestive. And that's really it. Yeah, I won't ask you on every example. I really just need you to know what mesothelia is, endothelia is, and simple squamous epithelium is, just like we learned in lab. Everything you learned in lab will keep you safe for the lecture exam, but you do have to add these two here. They will come back to haunt us. Next, we have stratified squamous epithelia. This is where we're going to have a tissue where we're going to encounter a lot of mechanical stress, friction, or even chemical stress from the external environment. Remember, stratified implies many layers of cells, and these are flattened. And these are the ones that I meant will have those desmosomes. Desmosomes anchor them next to each other very well, and that's why your skin is waterproof at the end of the day, because tight junctions and desmosomes keep it waterproof. Doesn't allow things to just go down there through the cells. And you find stratified squamous in two forms. What were they? Keratinized and non-keratinized. Whoa. Uh-oh. I think, sorry, I betted the wrong page somewhere. Yours probably doesn't have this. It's just mine. Yes, here we are. Okay, and so your next page should look like this. And so you see keratinized, you see it on the outer skin because it produces a protein called keratin inside of it. And then it also exists in a non-keratinized form, which can be found, again, in places of high friction: anus, vagina, esophagus, throat, pharynx. And that's what a pharynx is, your throat. And then oral cavity as well. What you know for lab will keep you safe in lecture. Know where you find keratinized versus non-keratinized. Cuboidal cells, we know that they make up glands, and so that's what it is. They function in secretion through the use of glands. An example now is exocrine glands. Cuboidal cells make up exocrine glands, and even the ducts of those glands. They are also capable of absorbing. When you look at a kidney in and of itself, like this is what your kidney looks like, but what it's actually made out of on the inside are a bunch of little worms that they're collecting, they're filtering your blood as it goes through here. And as it goes through there, whatever it thinks it's waste will go in here. And then eventually, whatever, it's just going to travel through this tube, and then eventually it's going to be urine and exit out the ureter, out the urethra, and it's going to be exit out as urine. But before then, if you examine these tubules, the kidney tubules, they look like this, like just a tube, and they're made out of nothing but cuboidal cells. And these cuboidal cells can also reabsorb, because your kidneys, when we say, oh, they can regulate volume, this is what we mean, is that water can actually exit through here, and your body's like, no, I need that water back. And so as it's traveling through here, it can actually reabsorb through those cells. So they will have, some of them might have microvilli, and they'll absorb solutes, ions, things like that you might need, but it can absorb. But remember, they can also secrete, which means goes in the opposite direction. Secrete, absorb. And so that's their superpower in a way. And stratified cuboidal exists. It exists but we just don't ask it in lab. Keep that in mind. In lecture, just need to know that it exists. And an example is going to be sweat glands and mammary glands. Sweat glands, we're going to talk about sweat glands tomorrow. And so this will come up again, stratified cuboidal epithelia. And so you can see they're exactly what you think. This is the lumen, cuboidal cells, and a stack of cuboidal cells right on top. And they're found in sweat glands. And you're going to have to know what a sweat gland is for integumentary system. Might as well just log it away, stratified cuboidal. One of those last little things to add. Transitional epithelia is going to do exactly what it sounds like. It's going to either stretch or relax Found in the bladder, nothing new to add. What you know in lab is good. Simple columnar. You remember we went over these in a few instances, and all we need to remember is that if it has microvilli, they absorb. If they have cilia, they move mucus. Yes, some columnar cells can have microvilli, and in other parts of the body, those same columnar cells will have cilia instead of microvilli. You don't need to know where and where just yet. We learned about goblet cells, and goblet cells produce mucus. They're considered unicellular exocrine glands. It just means like one cell gland. That's what this means. The gland made out of only one cell. And they secrete mucus. You already know this. And then pseudostratified columnar epithelia has cilia typically at the surface to move mucus. We'll discuss more details about that further down the line, but found in the trachea, and that's really it. Found in the trachea, I think is the safest one to remember. All right. And then for glands. We're going to cross off this page, entire page. Okay. Let's cross it off. You won't need it. And just keep in mind that it's useful in case you move on. Your notes will be a little more complete. But we do need to learn 3 categories of glands. Merocrine gland is the exact same thing that you've already learned, where the cell will produce, nucleus will command the cell to produce contents that will make mucus, that will secrete, and so it will go to the Golgi apparatus, and through the process of exocytosis, you will release substances onto the in the apical direction. So that's a regular form of secretion, merocrine secretion. It's just creating the product inside, and it travels to the apical side, and then releases out to the surface. And it produces mucus. And mucin now, I've said that we were going to come back to mucus and mucin. Mucus is just mucus, water plus mucin. If if mucus dried up, whatever's left, the contents in that liquid is called mucin. But when it has water, we call it mucus. That's the difference between these two words, mucin and mucus. It's like an equation, right? Mucin plus water make mucus. Without water, it's just mucin. And then we have apocrine. And you want to sort of use these first letters to remember the function of it. A piece of, like that, a piece of. Because these cells secrete in an entirely different manner. When they make their contents, they place it on the apical side. They don't let it out yet, though. They don't let it out into the lumen. They'll actually just put it in the apical side of the cell, and then it pinches off. That's it. That's why I say a piece of, because a piece of the cell comes off, and this is the secretion. This is actually what's going to be secreted out. And then it regrows, and then it happens all over again. And so that's an apocrine secretion. Holocrine, let the word hollow sort of sounds like whole, right? Like the word whole, like the whole cake, the whole something. And so holocrine, or entire, here like Latin, hollow, so kind of where it comes from, hollows. And this one, it's like a kamikaze cell, in a way that they sort of all burst. The cell bursts and kills itself in the process, but all its contents get released when the entire cell bursts. And that's really all the 3 secretions that you'll need. We'll see actual locations of them tomorrow. We'll discuss that. Like sebaceous. These are for your hair, if you want to just get a jump on that. Sebaceous glands are the ones that secrete oil onto your hair. And do not forget, endocrine places things into blood vessels typically. We're just going to keep it simple. Blood vessels or in the fluid outside so that it travels wherever it needs to. Typically, it ends up wanting to go to the blood vessels. And exocrine, right out of the ducts of those glands. Mm-hmm. Yes? Oh, yes.
An example of endocrine and exocrine? I already forgot. Like endocrine is pituitary gland, right?
Yes, exactly. Like endocrine would be like pituitary gland, the testes, ovaries, thymus, the pancreas, all those structures that we learned about on the endocrine organ system. which that just reminded me of something. I'm going to cut off one last thing from the first packet for all of you. And then muscle tissue. There is nothing new here to add that you don't know from lab. Cardiac muscle is found in the heart. Skeletal muscle is found on the bones. Skeletal muscle is multinucleated, has striations, and one thing to add actually is that skeletal muscle is voluntary. But we kind of knew that, right? We can move our skeleton by moving our muscles. The cardiac muscle tissue is going to be found in the heart. And it is special because it has intercalated discs. And we just learned what those intercalated discs are. They are gap junctions. They allow this cell to communicate with the one right next to it. This is why the heart beats as one, because these serve as a way to send the electrical impulse that the brain sends for it to beat, and it sends it to the cell really fast because of these gap junctions. In our lab, we've known them as intercalated discs. I also simplified the lab exam that you just have to know it as cardiac muscle cell, but as we progress further in the semester, you'll hear me say the word cardiocyte, implying heart cell. Nice and easy. And then smooth muscle found in the digestive tract, and both 2 and 3 here are non-voluntary. You do not control it. You cannot move the passage of your food through your digestive tract or beat your own heart voluntarily. And then nervous tissue. And nervous tissue, we have our neurons. We have our neuroglia. These are the assistive cells, the supportive cells, and the neurons are the ones that carry the impulses. Neuroglia come in different forms and have many different functions. All I really care for you to know is that they support neurons. This is a little bit extra than I want you to have, just it'll help for the future. And so just supporting cells, as written above for you, supporting cells. We know to find it in the brain and the spinal cord. And then finally, some structures that are attributed to the neuron are going to be the dendrites, axon, cell body, but the only one you need to know is cell body. We will come back to dendrites and axon further down in the semester, trying to save the space in your brains for the stuff that you need. So just the cell body, be able to identify the cell body just like in the lab exam. We will come back to axons and dendrites further down. And so back to connective. All right. So as we look at this pie chart here, your entire body is made out of only 3% epithelia. Only 3%. All those simple, stratified, transitional, squamous, columnar, those stratified, simple, all of that only makes up 3%. Only makes up 3%. The rest, only 2% is nervous tissue in the brain, spinal cord, and all the peripheral nerves. And then 50% is muscle tissue, primarily more muscle, like skeletal muscle more than anything. And then connective tissue is going to be the next major bulk, and that's the chapter where we end up, or we're going to keep for lab today. And something to add. When we look, and let me see how I organize these notes. I don't remember. Okay. All right, so what I want you to add to your notes, is there's this portion here. You'll see this fibroblasts and fibrocytes down here that I wrote right here. You want to write that down and bracket it in there. Fibroblasts, fibrocyte, except adipocytes. You want to make sure you write that too. And I'll explain what it means in a bit. It's too big. And I put a little L there because those are the ones that you are supposed to know in lab. That's the one we're going to go over today. I know that in lab number 2, this is what we're going to do today. We're going to go over areolar, adipose, dense, and elastic. And for those of you that already had lab this morning, we didn't do areolar tissue. We didn't do dense irregular. Those are removed from the lab to make it a little bit more easier to intro it and start it. But otherwise, these are other tissues that are made by fibroblasts or fibrocytes. And so for lab number 2, what this means is that these cells, or pardon, when we talk about connective tissue, it's a lot different from when we discuss epithelial tissue. Epithelial tissue had stratified, right? It could be stratified in layers, and they're tight together with tight junctions, gap junctions, and now you don't care about any of that. That does not apply to connective tissue at all. You want to sort of hold on to these cells as being their own little islands, like little spiders, and they produce their own little webs, or they produce their own secretions outside of the cell. And then they push each other around through their own secretion. They're sort of just around and then just creating the material and just pushing each other around. And you'll notice that in my little illustration here, there's a lot of space here. They're not directly attached to each other like the epithelial cells are. And so that's what you're going to see as a theme here, is that these cells are their own little islands or their own little entities, and together their secretions create an entire tissue. We'll get into little details on that soon. We'll see that there's a fluid connective tissue, and blood, for example, the one flowing within your blood vessels is considered a blood connective tissue. And then this, you're going to have to write this down here, cartilage. Remember that our word chondro was Latin for cartilage. So this means chondrocytes are the ones that produce cartilage. There are 3 cartilage in existence, and they are produced by chondrocytes. All 3 of them are made by chondrocytes. Chondrocytes. And then bone is made by osteocytes. Osteocytes. And then this word here is blasts. I know it's hard to read. Blasts. Oh, sorry. Osteocytes and osteoblasts. All right, let's get into it. Now, some properties. This image can be a little exhausting, but just keep in track that it's one image above, or one set of facts here, a second set of facts here, and I'll clarify. This is something you need to know. These are the 3 fibers in existence, and if I were to rank them, this one would be first place, second place, third place, as far as strength goes. Collagen is the toughest fiber. Elastic is the second strongest fiber, and also a little bit very flexible. And then reticular fibers, this one you don't need to know in lab. You'll see later today. We don't go over it. But reticular fibers exist, and they make a mesh network, like a mesh, a net, like a fishing net in a way. Now, we're going to get rid of some stuff here, so don't panic on all these terms. We are going to get rid of everything on the right-hand side. Lymphocytes, neutrophils, eosinophils. We're going to learn this all later on semester. There's no point in going over it now. There's enough to worry about. So you can cross off everything on that right-hand side. No wandering cells. And all it implies is that these cells are swimming through this tissue. But that's a complication I really just rather talk about after you know the tissues. And again, the key one that I care for you right now is fibroblasts or fibrocytes. I want you to know adipocytes. We'll see why in a second. And then melanocytes will be important for tomorrow. And I'm going to put skin. Talk about it then. And then the rest of it, you can get rid of it off this slide. How do you here? This, we'll get rid of it now, get rid of it now. Yeah. And we'll come back to it at an appropriate time. And now, what the heck are we looking at? When the star of this show, the star of this tissue, this is called areolar tissue. This is a type of tissue. And this tissue is made by the cell called fibrocytes and fibroblasts. These are the cells, and I'm trying to follow my lines here. This is going to be a fibroblast, and this is going to be a fibrocyte. And so these cells secrete fibers. They secrete collagen, they secrete elastic, and they secrete reticular. If we examine collagen, collagen is this thick, larger one. Elastic is this thin, hair-like one. And then reticular always looks like a little net. You're not really going to see it in lab. This is probably the second place that you're going to see it throughout the semester. And so this tissue is typically known as being packaging material that is just surrounding organs. It surrounds the organs. And so I use the packing peanuts analogy. You know when you get a very important package and it comes with the little foam, little peanuts that's to protect your package? It's a packing material, a packaging material. And then some last bit of terms in lab number 2. You're going to become a little more acquainted with this when we get there. But every single connected tissue we're going to talk about, all of these that were in the previous, all these that are in these boxes here, and the boxes, they are all characterized, or they all have a specialized cell. This first tissue that I mentioned, areolar tissue, its special or specialized cell is fibrocytes and fibroblasts. They produce extracellular proteins. They are these 3 up here, the 3 fibers mentioned above the photo. The collagen, elastic, and the reticular. These are the extracellular protein fibers. And now another word, matrix. Matrix again means area. So if you look at just this one cell, right? This is the inside of the cell, its nucleus, all its organelles are inside. And the space outside is the extracellular matrix, or simply the matrix. And so when we say the matrix, it means that there's stuff outside of that matrix. And in this space, you have to imagine that it's fluid, right? Like there's in the body, there's just fluid. There's not empty space. This is like all these cells. I know these are wandering cells. These are cells that float around through the tissue, through all these protein fibers, and they're involved in completely different organ systems like white blood cells. These are all white blood cells. And so these white blood cells are navigating through all of this, and they're swimming like, would you imagine a fish tank in a way when you look at this photo? You're seeing a fish tank, and you're seeing those fibers. You're seeing all these wandering different types of fishes swimming around, but the star of the show, the one that makes and creates this entire space, is the fibrocytes and the fibroblasts. They're the ones responsible for making the entire fish tank in a way. And with that, that fluid in that matrix is called a ground substance. This you don't learn in lab. So this is new for everybody. That fluid inside that fish tank, ground substance. And then to finish up our class, let's finish up on adipose. Actually, let's do these 2. Now, reticular. This one's not seen in lab. And so where we find it is typically in the liver. And what it does is that it's made out, it's called reticular tissue because it uses reticular fibers. Pretty easy, right? Out of those 3 fibers, it uses only reticular fibers. Only reticular fibers. And so if we look at reticular fibers on our illustration, it looks like a net. And so that net becomes very useful in places where we need a lot of movement. And so we encounter it, or whatever, we see a network, it's called a stroma. So we see it in the liver, the kidney, the spleen. And so for all of you, all you got to know, the name, the reticular fibers, and the name, the stroma, which means network. That's all. Made of reticular fibers. A stroma is just the name of that network, of that net.
Professor,
Yes, go ahead.
is that going to be in our background tomorrow?
No. Only the ones that are in your histology packet are the only ones that are going to be on the exam tomorrow. And now for adipose, this is where we're going to end. Adipose is made out of what cells? adipocytes. This is the unique one. This is why I had you write in the very beginning of this little section here. I had you write except adipocytes or adipose tissue. Because all of these tissues that we're going to talk about, areolar, reticular, they're made out of fibroblasts. Adipocytes, or adipose tissue, adipose is made out of adipocytes. Adipocytes. It's the only one that's unique. And what they do is that they store fat molecules. This is what fat is in the body. Fat cells, or like the lard of an animal, just the fat of an animal, the fats in general, they're stored in these cells. These cells are very unique, these adipocytes, because they store lipids inside of these huge vacuoles. They don't have a cytoplasm. It's so small that it's honestly non-existent. They just have a huge internal container, and it stores lipids. It stores lipids. And it's found everywhere, and stores lipids. All right. And lab number 2, take a nice little break. Oh, and before I let you